Today's speaker is not actually, I mean, these are the ones that I really enjoy. She's not that much further ahead than you are, in a sense. You know, sometimes at the ISS, we have people who are at the ends of their careers. They have, you know, gone on to lead long and distinguished research careers. Our speaker today is at the very beginning of her research career, in second year of your PhD, is that right?
Yeah,
second
that's right.
year of a PhD, right? So gone through a degree, now doing a research degree, a research PhD, and in the second year. And that's a really interesting time because that's where you're really learning how the whole research thing works. She's working in an area which is fascinating and which is, I'm really looking forward to seeing how this one plays out. She's from a lab at the university called Sydney Bionics, biomechanical engineering, taking science and technology and not just applying it to medicine, but literally applying it to the body, you know, physically attaching to the body so that we can help people live better lives. Would you please make very, very welcome, in the usual fashion, Jacinta Cleary. Jacinta, welcome to the ISS.
Ooh, thanks very much.
Go on, all microphones on and give us a good morning whoop.
Jacinta.
Thank you for that very enthusiastic welcome. So thank you for that introduction. So as Chris said, I am studying a PhD at the moment. I'm at the University of Sydney and I'm in my second year of the PhD. And the area that I'm working in is biomedical engineering. So that's going to be the topic of our talk. So basically, I have already finished my undergrad studies. So I guess you guys will be getting into your undergrads in the next year or two. And I did my undergrad doing a double degree in biomedical engineering and also neuroscience. So my kind of plan for that is that I'm really interested in how we can apply technology to interact with the body, but in particular, interacting with the nervous system and any kind of nerve deficit that people can get from injury or illness and things like that. So by doing that double degree, I kind of was able to get both that biology background as well as that engineering background, where I can kind of join the two and try and make that, try to make that technology that can be applied to that system. Just a fun fact about me is that when I was your age, I actually applied for ISS and I didn't get into it. So congratulations to all of you guys. And I'm just glad that I get to kind of have this little small part of the program at this later stage of my life, since I wasn't able to get into it when I was your age. So anyway, so we'll get started. Okay. So biomedical engineering, you can kind of, I think you can kind of guess, but basically biomedical engineering is super, super broad and basically covers everywhere that technology meets healthcare. So I wonder if this can work, but in the chat, can people just write down maybe some examples of what you think biomedical engineering is? Do you have any ideas of what it is? Yep, so Pepper, genetic engineering, prosthetics, bionic limbs, lots of prosthetics, yeah. Enhancing biology, pacemakers, perfect. Modified RNA, so yep, genetic sort of modifications. Yep, so someone put CAT scans, MRIs, X-rays, exactly. Cochlear implants, we'll be talking about that too. 3D printing bone scaffolds, amazing. Okay, I'll close that. Perfect, everyone has kind of a bit of an idea of what we're talking about. And it is this very broad field that kind of ranges across medicine. We've got tissue engineering, medical devices, robotics, so prosthetics that can either be just mechanical, or you can actually have an interface between to read the nerves that still exist, and then your brain can actually control a robotic arm instead of it just being sort of static. And then it also goes into things like ethics and so we've got nanomedicine and bionics and programming. And if you're interested in biology or chemistry or physics and things like that, it kind of all comes together to become biomedical engineering. And there's basically no way that as an engineer by yourself, you could actually do this make any difference in this world, because you definitely need to have the input of clinicians and biologists and chemists and everyone to work together to kind of come up with these really interdisciplinary ideas, which I think is one of the most interesting parts about biomedical engineering, is that you have to be interdisciplinary. You've got to work with a lot of people to kind of come up with an outcome that's actually going to positively impact the lives of the people that you're trying to impact. So from all these things, obviously won't have time to talk about everything. So I've kind of narrowed it down to a couple of 3 different things, which I find are kind of in opposite ends of the biomedical engineering spectrum. And so I'll go over these a little bit. And then at the end, I'll kind of take you through what my actual project is with a little bit more detail. So we'll start with tissue engineering. So tissue engineering is basically I think someone mentioned it before. So it could involve things like 3D printed bone scaffolds. And basically what it is applying the concepts of engineering to build or rebuild tissues. And so the idea is that we want to, in areas of injury or illness, we want to be able to restore, maintain, or improve tissue function. And basically the goal of tissue engineering is actually to create something that will become part of your body. So the idea is for it not to be a treatment for an injury, but actually to be a cure and to kind of recover, like repair and replace the tissues and organs that have failed because of those sort of illnesses. So basically the motivation for tissue engineering is for times when the body isn't able to repair by itself. So in areas like, so the first picture there, we have a bone that has this really large defect in it. So normally if you break a bone, your body can kind of just repair by itself. You just have to immobilize the tissue for a certain period of time and the bone regrows. But in cases like this, so if this is either caused by trauma and the bone shattered and had to be removed, or if the person had a bone cancer and that had to be removed, it creates this defect. And when it's over, I think 2 centimeters, the body is no longer able to kind of branch that gap. And so that could be because normally the body kind of is sending lots of signals out to direct new cell growth. But when the defect is that large, those 2 ends of the bone can't talk to each other anymore. And so there's no way for the bone to actually regrow and cover that space. And so in that case, you kind of need to think of a different solution to try and help that bone regrow. There's other cases where things like burns, where if the burn is so deep, then the cells underneath stop functioning correctly. And so you get, the skin is no longer able to regrow and replace itself like it normally would. And then there's also this huge emerging problem where we have people that have illnesses and injury and things like that cause their organs to fail. And we've kind of perfected this idea of the organ transplant, but the demand for organs far outweighs the supply. So there's this motivation for tissue engineering to try and work out ways to reconstitute these actual organs and tissue to try and meet that demand. So what is tissue engineering? So basically what we have is a injured tissue. We make this concoction of cells, biomaterials, and biomolecules together, and then apply that to the material and get the healthy tissue at the end. That's the goal. Obviously, you can't just kind of throw anything in there. It needs to be the right cells for the job. We need to have the right environment made by the biomaterial. So things like scaffolds to support the cells, and also biomolecules. So those are like those signaling molecules I told you about, that tell the cells how to grow and where to grow. So if we go further into it, you probably heard about stem cells before. So we need to take cells to start regrowing this tissue. We need healthy cells. Sorry. And these can be harvested directly from the tissue. So if you're trying to regrow a liver, you can harvest them from a liver. You can get stem cells. from the body. So in your bones, there are stem cells, and they can get divided into certain tissues. Or you can get stem cell lines that are grown in the lab and cultured in the lab. The kind of best case is if we're able to take those cells from the person because that limits the problem with rejection. The rejection is basically where the body sees the cells as an invading object, and the body wants to get rid of them. So then the stem cells can't do their job because they're surrounded by and attacked by other objects. Then we have biomaterials. So we want to put the cells onto a biomaterial. And the biomaterial is the sort of supporting structure. And that's the material that we want to interface with the bioelectrical system. And that is what's going to replace the tissue or organ or function of the body that's damaged. So that can actually be a biological material, but a lot of the time it's actually a synthetic material that we make in the lab. And we kind of can modify that so that the strength and endurance is what we need it to be for the body. So there are a couple of factors that are really important for choosing the right biomaterial. The first one is biocompatibility. So again, like I said, cells can get rejected. If a material isn't biocompatible, then again, the body will start to reject that. So a lot of the time it's because the body is seeing that material is foreign. And the further away it is from a material naturally found in the body, the more likely you're going to have a problem. So we try to keep it to like carbon-based things. And so ceramics are really useful because they're quite similar to the components made up in bone. And so we got mechanical properties. So things like if we're replacing cartilage with a synthetic material, we want the new cartilage to have the similar mechanical properties as the natural part. So basically the main reason for this is firstly, if we have something that isn't durable, as durable as what the natural cartilage would be, then that's going to fail really quickly. And so then that's not very useful to us. There's also an area where if the biomaterial is too good at its job, so if you want to have a biomaterial to replace a defect in bone, but then that biomaterial is too strong, then what can happen is that the force distribution across the bone is like most of the force is getting held by that scaffold. And then the rest of the bone becomes weaker because it doesn't actually have to do as much. So the idea is that we need to find mechanical properties that match what we're replacing, because if it's too less than or too greater than, then we're going to have problems with keeping this equilibrium inside the body. Another important thing is degradation. So not all tissue engineering implants use degradation, but basically the idea is that the end product that we want is that the body has fully repaired itself. And so the idea of degradation is that we have a scaffold that the body can grow into and then start to repair itself. And then as it gets more and more close to being back to normal, the scaffold starts to degrade. And so at the end of that process, all that we have left is natural tissue from the person. And so it's completely repaired back to the way that it naturally was. And the final one is that it should be easy to fabricate. So basically, if we have to climb a mountain in the Himalayas to find an ingredient to make this tissue or make this biomaterial, then it's not going to be very reproducible. And we're not going to be able to actually upscale it to be able to be useful for people that actually need the treatment. So the idea is that we want to be able to make the product with available resources so that it can actually be used by a lot of people. So the next one is, so that was cells, that was biomaterials. So the last thing is biomolecules. And so what I said before, the biomolecules are produced by cells naturally. So cells like proteins, amino acid sequences, etc. And they basically release like signals that tell the body how to, like what kind of activity or function they should be doing. So if we have, so things like growth factors are used to kind of direct the cell growth and function. They can also promote vascularization, so the blood vessels growing into the biomaterial. And they can also attract cells into the biomaterial or into the scaffold. And so the idea is that if you put this scaffold into that bone defect, then these biomolecules can attract the bones on either side of the defect to start growing into it. So then the biomaterials can be added directly to the biomaterial, or they can be, you can kind of add other things to the scaffold to try and get the biomolecules to go into the scaffold from the cells surrounding it. So as an example, so this is made in our labs. This sort of research is happening in our labs at the University of Sydney. So basically they've created these 3D printers that can print a specialized ceramic that matches the density and the hardness properties and things like that of bone. They make it into this sort of scaffold shape because that allows the new bone cells to grow through the scaffold. And then basically they put it into a defect and then kind of monitor how it grows over time. And so with these sort of 4 components, biocompatibility, it's made out of a ceramic that closely mimics those in bone, so we don't have very much of a problem with rejection. The mechanical properties are specially formulated to make sure that it matches the bone and can withstand the strength that it needs to while it's there without kind of taking that pressure off the bone that will stop it from being strong. It also degrades over time, and that degradation sort of rate is really matched to how quickly the bone can grow. So the idea is that the bone on either side of this scaffold will be able to, the bone on either side of this scaffold will start to actually grow into the scaffolds within all of these little, the little holes inside the scaffolds. And then especially with the biomolecules inside it, so it's signaling to attract that bone to grow into it. And then as the bone starts to grow into it, the scaffold begins to degrade and the bone takes up more and more of this space until that scaffold is completely removed.
Jacinta,
and by having this,
can I just interrupt there for a moment?
Yeah.
Just for any future slides that do have things like exposed bone, maybe a little gore warning. There's just a couple of people in the chat saying, I'm eating right now.
Right.
Yes,
I'm
gives
sorry.
a little heads
I'm
up
sorry,
when that one's
everyone.
coming.
Yeah, it's a bit early for these sort of images. Apologies for that. I did have it in my brain to give a note beforehand, but I forgot. I'm sorry. Did that just disconnect for you, or was it just me? Wait, sorry, my
No, we're still good.
screen is okay. Cool. Yeah, sorry about that, everyone. So yeah, so then like finally we have the easy to fabricate. So 3D printers are becoming more and more well known and used and understood. And so by having a material that is able to be printed, it means that it can have these really wide applications, and we can kind of make the shapes and sizes match what the defects that we're trying to fill. So just as a sort of future concept, we're currently able to print things like bone, because this, like a ceramic, is doesn't need to be vascularized or anything like that. But what we want to get to the point of is be able to print entire organs so that we can actually fill that gap with the sort of organ transplants. So at the moment, this is very difficult because of how complex organs are. They're not just made of one type of tissue, they're made of many, many types of tissues, and they have many functions, and they have their own biomolecules and things like that. And one of the biggest issues is that if you want to print something that large, by the time, so you can print the bottom layers, and you can actually print using cells and start to build some tissue. But what happens is that as you get further along, the tissue at the bottom, there's no way for that to get vascularized, and there's no way for the nutrients that it needs to get inside the tissue. And so what happens is that if you want to try and build this thing, it starts to like, the bottom of it starts to like die off before you're able to finish printing. That's just one of many problems that I've seen in literature recently. But it's just where all of the groups are kind of starting to build towards this area. And at the moment, they've been able to create quite complex tissues in like a 2D sort of way, so flat on a petri dish. So the idea is, how do we get from that point to growing it up into 3D and getting this whole organ out at the end of it? Okay, so now I'll go through some biomaterials. So biomaterials are materials that are in the nanoscale. And because they're in the nanoscale, they can do some pretty interesting things because they can get to places that we can't normally get to. So the obligatory nanometer scale, so you can see the sizes of things in nanometers, and nanoparticles are generally in this sort of, in the 1s to 100s of nanometers is what size the particles normally are. And so the nanoparticles are actually so small that you can't actually see them in the light spectrum because they're smaller than the wavelengths of light or visual light. And to kind of make you understand how small this is, thousands of nanoparticles can wrap around a human hair. So some general characteristics of nanomaterials. So as I said, very, very, very small. And so the idea is that because they're so small, they can enter into cells because they're much smaller than the cells. They're actually more similar to the sizes of the organelles inside the cells, and similar sizes to the biomolecules that are used to send messages around your body. Because they can enter cells, they can target and interact with specific biomolecules. And so we can kind of turn on and turn off certain functions of cells using nanomaterials. And they can also kind of exploit gaps that are really, really small in the body to be able to access areas of the body that we wouldn't normally get access to. And because of their structure, they have a very high surface area to volume ratio, which means that their surface is, their surface is highly reactive, which means we can do some interesting things and put some nice signals onto the surface of them, and do some engineering to kind of get the nanoparticles to do what we want them to do. So what can we do with nanoparticles? So really, really small nanoparticles, smaller than 5 nanometers, will just get cleared out by the kidneys. So we want to sit in this larger than 5 nanometer range. And basically the nanomaterials, once they enter the bloodstream, will circulate until they reach its intended destination. So they can passively enter the body using gaps in blood vessels around tissues, or they can do it actively. So I'm not sure if you might have seen this in biology classes or anything like that, but if we have, like on the cell walls, you'll have these sort of receptors, and the receptors are like a lock and key. So you need to have the specific molecule to attach the receptor to turn that on and to do this whatever function. So that's the triangle going to the triangle, and then that releases this shape that then goes and does something inside the cell. So what we can do is by engineering the nanoparticles, we can kind of attach different molecules onto the outside of it, and then it can kind of selectively go and interact with different cell membranes and cause different functions to happen. And then they can also be loaded with things. So the nanoparticle can act as a vessel for other things. So inside the nanoparticle, we can load things like drugs, fluorescent tags, and genes that when it interacts with the cell membrane, it can interact in that way with the molecule. So the active targeted cancer drug delivery actively. So some cancer cells express lots of EGFR on their surface. And so if we modify the nanoparticle to have this molecule on the outside, we can actually get it to selectively attach to cancer cells and then load that, express that chemotherapeutic drug directly at the tumor. And so obviously the benefit of that is that conventional chemo treatments work across the entire body, and there's the problem that that starts to attack other parts of the body and you get these sort of really horrific side effects. So if we can specifically target the cancer cells, one, we can make sure that we get this really high therapeutic dose at the cancer, but two, we can stop that sort of widespread impact on the rest of the body. So then there's also this passive method where tumor vessels kind of attach and get a blood supply from your vessels, but they cause the blood vessels to become leaky. And so that kind of makes gaps in the blood vessels that allows the nanoparticles to basically swim past all the normal vessels and then leave through the leaky areas. And then that's another way that it can kind of target the tumor vessels. And then it can also do things like biosensing. So this is where instead of loading the nanoparticles with cancer drugs, we can load them with fluorescence, or there's some nanoparticles that actually are naturally fluorescent. And then same sort of deal, we kind of attach these surface markers onto the outside of the nanoparticles, and then they attach to different cells. And then when they fluoresce, we can see which cells are present that match what we wanted them to bind to. So things like we can target cancer cells and then they'll light up and you can see where the cancer cells are. And the idea of this is that we're working towards a system where someone can take this sort of treatment, and then we can, while the person's like just a normal live body in some kind of imaging mechanism, we can see if any of their body lights up with these nanoparticles, and we can do like really early detection of cancer cells, because since the nanoparticles are so small, it can detect really small amounts of these cancer cells. So the idea is that we can get really early detection and detection of really small amounts of cells in a cost-effective and non-invasive way. Okay, so final area. So I'm going through this very quickly, but you can ask me questions at the end if you want more detail about things. So this is my area, the implantable bionics. You probably have seen things like this before.
Okay, ready? Here we go.
But basically, these are the cochlear implants where people born deaf are able to start to hear again.
More. All right. How does it sound now?
Good.
Is it sounding better? Can you hear my voice now? Yeah? What about Mom's voice?
Can you hear my voice?
Yes.
Hey, Dawson. Dawson. Oh, you don't like it anymore. Is it scary? You hear the bird? That bird? That little tiny bird chirping?
Yeah.
Could you really hear it?
Yeah.
Could you ever hear that before?
No.
No? What else? Hear this? Yeah?
Yeah.
Could you hear that before? You hear those cars on the street? Yeah? You like that?
Yeah.
So I'm not sure if any of you have seen videos like this before, but watching things like this was kind of my motivation to go into bionics, because of the profound impact you can make on someone's life by kind of interacting with and restoring functions like hearing or seeing and things like that that have been lost to injury or illness. So these in the video are cochlear implants, so to restore hearing to people. So just a little bit of details about cochlear implant. So you basically have this part that you can see from the outside has a microphone that you can hear so that it records basically what's happening around you, and then processes that sound and then transfers it through this area into the implant inside the head. And so there's a matching part under the skin here, and then it goes down into the ear canal. So what we have on this side, this thing is called the cochlea, so that's why it's called the cochlear implant. And that basically is a organ inside your body, inside your inner ear, and that's what helps you to hear. And so arranged in this sort of spiral shape, you have these little hair cells, and they react to sound waves, the different frequencies of sound, differently. So at one end, the hairs will be vibrating when the sort of deep, low sounds come through, and at the other end, they'll be vibrating when the high-pitched frequencies come through. And it's sort of like a piano, so it all goes in sort of this like coordinated sort of way. And when those hair cells are triggered, it sends electrical signals to the nerve, to the auditory nerve, and then sends that to the brain. That's how you perceive hearing. So with the cochlear implant, what they do is they put, it happens when those hair cells are damaged, so they're no longer able to respond to sounds. So what they do instead is they put these electrodes into that sort of spiral region, and then that has about 27 electrodes along this line. And so it means that the microphone kind of records the sound around you. It gets processed into 27 distinct pictures, and that gets transmitted through, and the electrode stimulates the cochlea along its length to kind of correspond with the sounds you're hearing. In most of the cases, the auditory nerve isn't impacted. So then by directly electrically stimulating, what you're doing is stimulating that nerve, and so it still sends that signal to the brain. But obviously, 27 pitches is very different to the millions that you probably have naturally. So the hearing isn't exactly how you would normally hear it. It's quite robotic, but it's still definitely much better than being completely cut off from the hearing world. And so a lot of the advances that we're trying to make in bionics is to miniaturize and improve this technology so that we can go from 27, which is the current limit to how many you can actually kind of fit in because it's so small, and try and miniaturize all that technology to try and add more and more and more of these electrodes to try and get as close to natural hearing as we possibly can. The same goes for bionic eye. So you have genetic disorders of the retina, where things like retinitis pigmentosa means that all of your peripheral vision starts to go away and it creeps further and further into the center of your eye. And then there's also things like age-related macular degeneration, where the center of your vision gets degraded. And at the moment, there aren't really any effective treatments to fix this sort of progression into blindness, even though there's millions of cases each year. And so as a little bit of background, so you have, these are basically the cells that you have going from the surface of the eye back to the nerve. And when you have these conditions, these cells that are the ones that interact with light and are stimulated by light and allow you to kind of send that electrical signal to the brain, those ones start to die off, and then you can't, that signal is lost. But on the positive side is that the cells that actually send the signal to the brain are still active. So again, we can begin to add this electrical stimulation directly to these nerves and get some perception of vision. So what we do is we basically, so this is what the bionic eye electrode array looks like. And each of these black dots shows a different area where it's going to create an electrical signal that will interact with the neurons at the back of your eye. And so the idea is that it gets implanted at the back of your eye as close as possible to the retina. And then that is going into like the focal point. And so then that can hopefully stimulate to start to recreate vision for someone who doesn't have vision. But again, we have millions of cells normally, and that kind of gets reduced to, in this case, so the Phoenix 99, which is the bionic eye that we're developing in my lab. In this case, we only have 99 points of vision. So the idea is that we need to kind of improve this technology so that in the future we can get as close to this the vision back as possible. And so finally, I'm not sure how much time I've got left, Chris.
Oh, you're okay. I mean, we're only coming up to sort of towards 40 minutes now or so.
Okay,
so,
great.
you got a bit of time and we're flexible.
Okay, cool. Okay, so the last thing I wanted to talk about, so this is the project that I'm working on. So it's the restoration of natural blink after facial paralysis. So basically, what we're trying to do is for someone who has had their face paralyzed by some kind of condition, we want to try and restore their ability to blink. So I'll go into this a bit more detail than I've done for the other projects. So firstly, so we're trying to interact with, our target population of people who have had damage to the facial nerve. So what does the facial nerve do? You basically have two branches of the facial nerve that go onto either side of the face. So if you have, and then the facial nerve is able to produce some of your facial functions. So like your facial movements, so raising your eyebrows, smiling, frowning. It has a little bit of function in your inner ear. It helps make tears by the blinking process, and sends some information about taste from your tongue to the brain. But definitely one of the main things that it does is that it controls the muscles that cause your eye to close. So if you imagine not being able to blink, if you've ever had a staring contest with someone, you can kind of understand how quickly you can get to the point where your eye starts to get really irritated, you start to have tears streaming down your face, and it kind of becomes almost unbearable after just a few tens of seconds, right? So these people are kind of experiencing this permanently for the rest of their lives. So, but not only that, not only is it uncomfortable, but they then have to try to recover the function of blinking, which is to lubricate your eye. And so they have to constantly be applying medication to their eye to try and keep it from drying out. And people blink around 16 times per minute. So that is a lot of sort of intervention you have to be constantly doing to make sure that you're keeping that eye healthy. And then if you don't manage to do that efficiently, then your eye starts to dry out, and you can start getting ulceration onto the eye. And if that is untreated again, then it can actually lead to blindness. So obviously, be able to blink is very important for your just overall health and even your vision and how you interact. Then there's also this other side of facial nerve paralysis, that when we have surveyed people with facial nerve paralysis, they've said that one of the worst parts of having facial paralysis is actually that they lose this sort of facial symmetry. And so their not able to blink in time, and your eye contact is what helps you kind of interact with the people around you. And so it's the first thing someone sees if you have a disability on your face, is that you're like, it's very noticeable. And so what happens is that strangers and acquaintances will come up to them and ask them, like, what happened to you? They'll ask these intrusive questions, like, what's wrong with your face, constantly, and get stared at when they're out in public. So these people start to self-isolate because they don't want to have to deal with that every time they go out into society. So basically it means that where it's not just the physical side of things that this disease is impacting, it's the person's entire life and quality of life, because they no longer feel like they're able to participate in their role in society. So if we look at how blinking normally works, so this is how normal blinking. So we have two different muscles. So we have one that holds the eye open. That's called the levator palpebrae superius. And that gets interacted with by cranial nerve 3. And so when your eye is supposed to be open, then this is constantly firing, telling your eye to stay open. And then when you want you to close your eye, that one stops firing. And then this other muscle called the orbicularis oculi, which is innervated by the facial nerve, by cranial nerve 7, then that tells that orbicularis oculi to contract, and it's a circle around your whole eye, and the whole thing contracts and causes your eye to close. In facial paralysis, obviously you lose that innervation by the facial nerve. So now what happens is that your eye is open, caused by cranial nerve 3, because that's still active. And then when you want to blink, nothing happens. So you kind of get this situation where when your eye is open, it actually becomes more open, because you don't have the muscle tone of this muscle kind of holding it as an antagonist to the open. And so your eye becomes more open naturally. And then when you want to close your eye, because this one still deactivates, your eye can kind of droop a little bit, but not very effectively. And you don't get that full closure, which can disperse the tears across your eye to keep it hydrated, as well as that sort of blinking helps the tears to exit the tear duct and produce more tears to hydrate your eye. So at the moment, there are a few different things that people have used as interventions to try and help people with facial paralysis, to help them with blinking. The most common by far is called gravity-assisted closure. And basically what they do is they get a little weight made of gold or platinum. So those are both biocompatible materials. And they implant it into the eyelid. And so the idea of that is that when that muscle that holds your eye open deactivates for you to blink, the extra weight in your eyelid will help the eye that's drooping down to just droop faster. And so I think it's effective at improving eye closure in about 60% of people. So it's not super effective. And a lot of the time people only get about 80% eye closure, which isn't really sufficient to improve that sort of tear hydration of the eye. The other problem is, so another thing is that because it's relying on gravity, if you're lying down, then you don't have that gravitational force, so that doesn't really work. Or if you're not completely upright, it doesn't work quite as well. And as well, because it is just relying on gravity, it is quite slow. So you'll have one eye blinking normally really quickly, and the other one will kind of droop slowly closed. So the main problem with this is that it doesn't address this aesthetic issue, and it causes an incomplete blink. Another method that people have attempted is electrical stimulation. The problem with this one is that we're relying on all of the sort of muscular structures. So electrical stimulation, you would directly stimulate the muscle and cause the muscle to contract. The problem with that is that you're relying on the musculature to all be intact, which isn't necessarily true in cases where the cause behind facial paralysis is things like tumor resection, where that muscle has to get removed, or parts of the muscle. There's also a problem where, because you're trying to stimulate this sort of large area, the amount of stimulation that you need to effectively close the eye starts to actually impact your pain receptors. And so there's not much difference between like the therapeutic amount of stimulation that you need and the amount that starts to cause pain. So this hasn't really developed towards clinical because of these sort of problems. Another method, a surgical method that's been used, is called muscle transfer. And basically what they do is on your temple. So if you've ever noticed when you're chewing, your temple kind of goes in and out. Those are muscles that kind of help you chew, but they're not really very useful anymore. They kind of duplicate musculature. You don't really need that musculature. And so what people do, or what surgeons do, is they will they'll take part of the this temporalis muscle and they'll dissect it from the bottom, and then they'll move it across your eyelid and put it around your eyelid and basically recreate the muscle function, the muscle that's lost. So by doing that, you then have an active muscle in your eyelid, and that muscle can actively contract and cause the eye to close. But the main problem with that is that the nerve that is causing that muscle to contract still remains as the original nerve. So if you're using the temporalis muscle, that contracts when you chew. So it means that every time you chew, your eye starts to blink. And every time you want to blink, you have to remember that you need to clench your jaw. And so it's not very natural. And it's also quite uncomfortable for the people that have this. So basically what we're trying to do is we want to correct these problems that we have with the current interventions. And the way that we've come across that we're trying to approach this is using an electromechanical implant. So basically, what we're doing is creating something that can actively work with the body and create this eye closure. So what we do is we get a sling that's made out of either tissue from the person or it can be made out of like silicone or suture wire or anything like that. And it gets implanted into the eyelid. And so it gets attached at one end at the bridge of your nose, and the other end gets attached to the end of the device. And so that part is what we call the plunger. And that is basically a cylinder of magnets. And then we have inside the device, we have an electromagnet, which is basically a coil. And so what happens is that when we want the eye to be open, the device will be charging, and there's like a capacitor or a large battery inside the device. And then when we want the eye to close, then that battery discharges really quickly through the coil, which creates a magnetic field and pulls on that plunger and on the sling, and that closes the eye. And then because that muscle for keeping your eye open is still active, once that happens, that muscle then reopens the eye. So just to give you another example of it. So down the bottom is the charging and discharging of the battery. And then this plunger is magnetic, and it pulls back on the eyelid and closes the eye. Oh, sorry. Okay, another mildly gruesome image for anyone who doesn't like that. Maybe close your eyes for a little bit until we get through the next slides. But so basically we have, this is the device in action. Oh, no, I don't want to show that one yet. Okay. Slides aren't working for me. So we have, this is a sheep. So sorry, it's not working for me. Okay. So the one on the left is the actual function of the device. You can see how quickly it's moving. Okay. You can see how quickly it's moving. It's attached to a spring just so that it returns back to its original position. But so basically what's happening is that when the device is in this position, that's when the eye is open. And then when if it's going to play, and then it pulls back really quickly and it causes that blink. So blinks only last about 80 milliseconds. So it needs to be a really quick movement for it to kind of replicate what a blink should look like. And then this video, which has already gone through, this is from one of our studies that is working towards animal studies and then clinical trials. And so basically what is here is that this is a sheep. The nose would be here and the ear is here. Oh, wait, sorry, you can't see my mouse. The nose is at this end and the ear is at this end. And we have the sling is implanted into the eyelid, and then the device is sitting at this end. So when the device is activated, it pulls back on the eyelid and causes the eye to close. For anyone that was closing their eyes during the gruesome part, that's gone now. So basically what I've had to do, so you can see this sort of GIF going through. These are all of the design iterations that I've gone through to get to the point where we're at now. And so it's been a lot of, basically the shape changing is to help it match better with the biology. So to fit into the shape of the skull of the sheep, it needs to be in this shape so that it can fit comfortably. I'm also using so I've originally started printing out of resin and then eventually got to the point where we're 3D printing out of ceramic. And so what we mentioned before is that ceramic is a biocompatible material. And so by having this material, the body isn't going to flag that as a foreign material too much and will allow it to be in the body without having that inflammation. And then we seal everything up so that the plunger is moving in and out of the device, but because we're using electromagnets, we're acting on the plunger wirelessly. So the plunger is interacting directly with the body, but all of our electronics are inside the device and they're hermetically sealed. So basically what that means is that none of the body fluid, anything like that, moisture can enter the device, which would ruin the device. And none of the electrical charge or any of that sort of components inside the device can exit, which would cause problems with the surrounding tissue. So once we got to that point of the study, we then did testing for things like making sure that everything is hermetic. We had to sterilize all the materials to make sure that it didn't have any bacteria or anything on it that would cause an infection once it was implanted. We also did studies to see how much force was required to cause the eye to close. And then we made sure that was kind of our target for the device, to make sure that it would be able to, at minimum, hit that line, which we found was 570 millinewtons. And then our device can kind of create a much wider sort of band of forces. And so the idea there is that once the device is implanted, we can actually modify how much power goes to the device, and that will directly correlate to how much force the device produces. And so that means that we're able to tailor the device to each individual and make sure that this device is able to create a force that can fully close the eye on each person. And then as well, once the device heals, maybe there'll be changes to how much force is required. And so again, with a clinician, the clinician can then go through and modify that force over time to make sure they continue to maintain this full eye closure. Something I didn't mention before as well is that at the moment, our device is just triggered every 5 seconds. So open for 5 seconds, closed for, trigger the device to blink, and then open for 5 seconds again. The future version of the device will include a neural like sensing electrode. And so that will be attached to the opposite healthy eye, because facial paralysis is only impacting half the face. And that will tell the body when you want to blink, and then will trigger the device to blink to create that synchronized sort of eye closure. And then this picture on the right is showing our first animal surgery, where we implanted just a passive device, so one that wasn't going to activate. And that was just to confirm that our biocompatibility and hermeticity and all of those sort of properties, as well as the fit and the surgery and all those sort of things are working, so that we can so that we can make sure that the device is at the stage we want it to be before we then embark on a larger study where we can see how things are actually working. Over COVID, when we were all in lockdown, I developed this software as well, which I will use once the device is actually implanted. But so basically, what it's doing is I've recorded my eye on my phone, and then the software is increasing this contrast to detect the edges of my eyelids, and then it's taking that information and detecting when there's a blink. And then from that information, I can do things like extract the velocity of the blink and the displacement of the blink. And then I can compare that to what the device is causing and kind of get this really well-rounded idea of how well the device is matching natural blinking and how close we are to getting to that point that we're trying to reach where we have this natural blink. On this side is a recording taken from a sheep, but that was a cadaver sheep, like the recording earlier. So, which is why the eye isn't opening quite as we would hope it would, and it's not the same speed and things like that. But it was basically proof of concept to show that the device, this software that we were creating, will work in both humans and sheep. Okay, and that's all my presentation. I see that I've got many questions in the chat box. So thanks everyone for your attention, and let's get to these questions.
Thank you so much, Jacinta. Yeah, there are just a few questions. And I mean, it's always interesting to see the questions come up and then like half of them you then answer on your next slide. Why don't you stop sharing your screen so that other people can come on as well? There are lots of questions that haven't been answered though. And I think, why don't we start with something which is quite near to the end of what you were talking about. There are some questions which are very specific to that thing that you just showed then. How does that work? And then there are much sort of more meta questions. So let's go with one of the specific ones, one of the almost very last ones. Lily Hackett, if you're there, you got one which a lot of people then went, yeah, how does that work? Lily, put your microphone on. You got a question?
So how do you stop the blinking movement when the person wants to sleep?
Yeah, like
That's
if you've got this
question.
thing making you blink all the time, how do you turn that off? How does that work?
So if you saw from the cochlear implant, what the cochlear implant sort of looked like a little while ago, you have this sort of internal component and the external component. And the external component is the part that's actually producing sending the power across and charging the device and keeping everything working. So there kind of is two parts to this. So if that external part is removed because you're going to sleep and it's more comfortable that way, you'd remove that external part and then you're no longer providing power to the device, so the device doesn't work anymore. Obviously, the other side of that is that if the device is causing your eye to close, then what happens when you're sleeping? Because obviously you want your eye to be closed while you're sleeping. So in that case, a lot of people that have this condition would normally just do something like tape their eye closed or have some kind of other mechanism to make sure their eye was closed while they were sleeping. So that would be kind of addressed as a separate thing to the device. A good question, yeah.
Cool. Thank you very much. Okay, so on to the next one then. This is sort of working our way back through in a way. You did show, you know, a bunch of sort of hypothetical things and prototypes and so on, but then you also did show actual animals and, you know, medical operating rooms and things like that. So Caitlin Tan has a question about ethics. Caitlin, do you want to throw your microphone on and ask your question?
What ethics do you consider when testing the product, and like what regulations do you need to pass to like get it to test on humans, like for your next step?
Yeah, that's a great question. And that's something that we learn very consistently and in a lot of depth throughout the biomedical engineering degree. Because obviously, anytime you're working with medical products, you need to make sure that you're not causing harm by what you're doing. So specifically for my project, we, to be able to do anything using with animals, we have a animal ethics committee at the University of Sydney. And we have to go through this really long process to kind of prove that our product works, firstly, and then also that we've done everything we possibly could to bench test it to make sure that that is all validated before we get to the point where we can actually do anything in live animals. So a lot of my work has actually been on cadavers, because then that means that the more that we can prove things in cadavers, the less we actually have to use live animals. And so we're only now getting to the point. So this project's been running since 2014. And I started working on it in 2020. And so we're only just getting to the point where we can actually implant it into a live animal and feel confident that what we're doing is actually something that's going to progress the science and isn't going to cause undue harm, but also giving us as much information as we possibly can get to make sure that we can progress through. If we wanted to go from that point to human studies, there are many, many more regulatory hurdles that you need to go through. And especially you need to have really solid data, both from the bench and from animal studies before that, to make sure that by the time it gets into humans, we know 100% it's going to do exactly what we want it to do. And all you're trying to work out once it's in humans is sort of the small things like how much force is required, like how is it specifically interacting with the human body compared to how it's interacted separately.
Yeah, I mean, it's a really interesting thing about doing research at university that involves anything alive, but particularly humans. You learn a lot very quickly about the whole ethics thing. And, you know, I used to do research on physics education where we would give surveys to students, and just asking human beings questions requires an ethics committee.
Yeah.
So let alone actually sticking things into their body. That's a whole other thing. Listen, speaking of sticking things into their body, are you okay to take a question about, I know it's not your specific project, but when you were talking about the bionic eyes and bionic ears, a bunch of questions came up. So are you happy to take a question about that? And we're totally fine for you to say, yep, I don't know. Go and find someone who's working on the project.
I know much more about them than I do about the tissue engineering and that.
Okay, fair enough.
Throw them at me.
So Rio Tano, if you're out there, do you want to throw your microphone on? You had a question about the bionic eye.
Okay, can you hear me?
Yes, we can.
Yeah.
Well, are there any differences of visible colors between the real eye and the bionic eye?
I mean, this is kind of getting into that question of, do you see the same red that I see? Except that this is now really in the regime of, but you've got a bionic eye. Like, is it the same? What level are we at?
So I wish we have a simulation of it. So firstly, the bionic eye isn't able to differentiate between your red, blue, green cones in your eyes. And those are the things that give you color vision. So it wouldn't be able to kind of give you back the ability to see, oh, that's red, that's orange, and things like that. You'd be seeing completely in black and white at this stage. And then the other thing is that your vision is going from millions of cells to, in this case, 99 at the moment. So if you're imagining your computer screen, it has all these pixels. If you decrease the number of pixels to 99, you're going to be seeing a very degraded sort of version of what you could potentially see if you had more cells. So at the moment, the greatest benefit of the bionic eye is to kind of give people that have no vision the ability to start to become more independent. So a person with the bionic eye won't be able to like recognize faces necessarily, might not be able to like, so you would normally have like a camera that is recording what you're still seeing around you, and then that gets interpreted and then sent to those electrodes, and it tells the electrodes which ones to kind of fire and what you should be seeing. So depending on like, so we have like a VR headset in our office that kind of shows what it would be like to have the bionic eye. And if you're just looking at a screen, you can't see anything. But if you put the camera really close to the text, then you can start to read with it. So it's kind of, but then if you're wearing it and you're walking around the office, you go from not being able to see it all to being like, that's a wall, that's a chair, that's a table, the door is there, sort of things like that. So it means that a person that was before completely blind might be able to kind of walk around more independently without being worried about their environment as much.
So it's sort of moving in the direction of recreating what a sighted person has, but you can't think of it at the moment as, oh, I've got a fully functioning eye.
Exactly.
It's I have vision of some kind.
Yes.
And that sort of leads to the next question, which is from Luke Palmer. Luke, are you there? You had a question about the limiting factors in bionic implants.
I am. So I'm trying to remember which one was. So like with bionic implants and all that stuff, is it in terms of improving, the technology, is the limiting factor like we need smaller sensors for bionic eyes or better implants, like better materials or whatever? Or is it like we need to know more about how organs work? Like which of those is, or is it a bit of both? Like,
yeah. So I think for the eye, we know a lot about how vision works, at least inside the eye. So talking about how vision works in the brain, completely different. But in the eye, we kind of know there's all of these photoreceptors that are interpreting the light and kind of sending them to the nerves so that you can see. And so in that case, the limiting factor pretty much is the ability to get the electrodes small enough. At the moment, the way that so that electrode array that I showed on the bionic eye slide, that's actually basically a foil of platinum that is laser cut by a $750,000 laser. So like one of the top sort of rated things that we could get, and that is cutting as small as it can to try and fit as many electrodes as we possibly can onto this foil, and then we attach it to the rest of the circuitry. So at the moment, this 99 electrodes is at the edge of our capacity of being able to create. But with everything, computers used to take up entire rooms, So it's like the further that technology kind of improves, the more that we're actually going to be able to do. So hopefully in the future, the possibilities will be endless for what we can achieve.
Yeah, I mean, if you look back at the first transistor that was ever made, the first electronic transistor, it was huge. It was this big chunk of stuff that looks like it's made out of Play-Doh, and now we have trillions of them on a tiny chip in your phone, right? So the progress of technology is in that direction. Listen, Jacinta, we have time for probably just one more question, and this one's going a little bit meta, but I think like I had a choice of a lot of different ones for this final question, which are all around, these are all high school students looking to figure out what they do next. And you are a young researcher, so they're all keen to know what you know. But I think I'm going to go with Jake Noble, if you're there. You had a question fairly early on, which was kind of an existential question. So if you're there and you want to throw your microphone on, you can ask that one.
How do you know if researching is something you want to go into in the future? And I guess also how do you know what area is good for you?
Mm-hmm.
Jacinta, do you even know if research is something that you want to do in the future?
It's a great question. So for the first part, one part of the question is, how do you know which area that you want to get into? I think I was quite lucky because I had no idea what I was going to do. And then I went on a science camp like you guys are doing, and I was introduced to biomedical engineering, and I thought, oh, that's pretty interesting. I'll kind of have a look into it. And then I went to the University of Sydney Open Day, and they just had like a room where they had one of the professors in there that you could ask questions. And they had this big screen on with like a video that was showing a person that had this really severe muscle, I think it was called muscle dystonia, which is basically where your muscles are firing without you telling them to. And so he was growing up completely normal. And then by the time he was about 8, he started to have this issue where his body would just spasm uncontrollably. And so then by the time he was in his late teens, he wasn't able to walk anymore because his body just wasn't doing what he wanted it to do. So then this group developed a brain implant that basically stopped all of those aberrant signals from the brain and allowed him to have control over his body again. And the end of this video shows him just like riding a bike again for the first time in his late teens or 20s after being bedridden for years. And so I saw that, and I'm just like, that is what I'm going to do. And so from there, I kind of had this really clear path of doing biomedical engineering and neuroscience. But in terms of knowing whether or not to do research, that's definitely a very individual sort of thing to think about. I think at university, so if you do engineering like I did, there's a requirement to do a thesis in your final year. And the thesis basically is like, instead of doing one of your subjects, you do a research project. And you have a supervisor that's one of the professors and things like that. And you kind of get like a taste of what research is like. And so I did that and I enjoyed it. But I still wasn't sure. And then I worked, after I graduated, I worked in a lab for a couple of years, not doing research, just kind of got a research assistant to helping out in a lab. And I had my own project and I was kind of working on it, but it wasn't really officially research. And then my supervisor was just like, hey, do you want to do a PhD on that now? And so I kind of fell into it and kind of just started doing this. But I think the big benefit of doing research is that you have a question and you're the only one who's kind of looking at that question in the way that you're looking at it. And you get this freedom to kind of go in all these different directions and make your own path. And in engineering as well, I'm just like, we make our own equipment to build things and then it doesn't work. And then we go in a different direction and things like that. So I find research really exciting. Whereas like working in a company, I might not have as much freedom to kind of go out and break things and then fix them and work out what I'm going to do. So I find the whole research area really exciting and really interesting to work in.
I think you've touched on something there which is really, really important. And I would say any of you watching this who are thinking of going on to university, and it's probably most, if not all of you, there are loads of opportunities at university to try out different things like research projects or an industry placement or whatever it might be. And I can also tell you that there are loads of those opportunities that don't get taken up because students don't know about them or don't explore them. So if you're asking those sorts of questions, then when you get to university, look around and see what's on offer because you'll have a chance to try stuff out. It's out there.
Yeah. That's so true. So as well as doing engineering, it's a requirement to do an internship at some point in your degree. But there were some people in my class that were doing internships from their second year. And so by the time they graduated, they'd gone and done a few weeks at a bunch of different companies and as well worked in labs, done small research projects. And it was just because they kind of asked people, And then by the time they finished, they had this great idea of what they wanted to do because they'd actually kind of tried everything. And there's also at university, there's societies that will host things like industry nights and panels and things like that, where you can talk to people from industry, talk to people doing PhDs, talk to other people, and just kind of get an idea of where they're coming from and what their kind of answers to that question is as well.
Yeah, no, there's just so much good stuff that you can do. So go and check it out. Listen, we need to stop there. Jacinta, thank you so much for your talk and for showing us not just your research area, but your particular really cool area of study. So can I get everyone to throw your microphones open again and give us a whoop of thank you to Jacinta Cleary.
Thank you.
Thank you. It was awesome.